Good day, this is Professor Will, CBMD PhD. Today's Thursday, October 22nd, 2020. It is 8:29 and 5 seconds p.m. Eastern Standard Time, and this is a continuation of my microbiological research, so let's hop to it. Early attempts to devise truly anoxic culturing techniques included the use of deep agar shake tubes, gas with hydrogen, the use of pyrogelo to remove oxygen-sealed tubes, and the isolation of colonies by picking the capillary pipettes. Still, the only anaerobes isolated until 1940 were spore formers and several non-spore-forming bacteria of clinical importance, while identities of majority of the anaerobe prokaryotes in sediment, soils, and gastrointestinal tract remained unknown. This was part due to the fact that techniques were insufficient to reach the necessary anaerobic conditions, but also to the fact that the media used were not habitat simulating. The Hun-Gate technique was very successful, and has been widely adapted by microbiologists from various other fields of dentistry, sewage, and sediment microbiology. Many soil bacteria were studied in pure culture after their discovery to examine basic metabolism and metabolic capacities. The researchers emphasized that the conclusion drawn from laboratory studies on the behavior of bacteria in natural habitat may be misleading, because laboratory strains might only be artifacts, i.e. selected by the chosen nutrient medium, temperature, aeration, and agitation. Consequently, researchers already warned against the use of bacterial strains from culture collections as typical wild-type cells, and together with some of his students, designed methods to study bacteria in situ within their natural habitats. Research concepts on the soil microbiology on the differentiation between autochthonous and allochthonous populations in the soil are the basis of relevant current research. The majority of physiological investigations have been limited to pure laboratory cultures. However, Koch's methods may also have hindered the understanding of the microbial interactions as they occur in complex natural microbial communities, e.g. in soil. In fact, interactions between microorganisms under natural conditions may lead to transformations that are unknown from pure cultures. The shortcomings of pure culture techniques are illustrated best by the interspecies hydrogen transfer described, as well as by the inhibition of growth of Micrococcus culture by chance, which led to the discovery of antibiotics. For an in-depth understanding of microbial physiology, microbial transformations therefore have to be studied, also in natural mixed microbial communities. In media of appropriate composition, and in the presence of suitable substrates, bacteria multiply. The rate of growth is dependent on substrate concentration, temperature, pH, and osmolarity. Can be accessed from the increase in bacterial cell number or in bacterial biomass. This distinction has to be made since under certain conditions growth is not completely balanced, and the increase in biomass is not paralleled by an increase in cell numbers or vice versa. As an example, cells in the early stationary phase can undergo reductive division, whereby cell numbers continue to increase while a constant overall biomass of the culture is maintained. Upon inoculation of bacterial cells into fresh growth medium, a period without growth is frequently observed, the so-called lag phase, during which adaptation of the cells occurs by e.g. synthesis of cellular enzymes. Afterwards, a phase of exponential growth occurs until cell growth is limited by substrate availability and by accumulation of toxic metabolic products or unfavorable pH stationary phase. The stationary phase is then followed by a phase of prokaryotic biomass reduction due to lysis of cells. If changes in bacterial cell numbers n are followed during the exponential phase, the specific division rate and the time generation are used for the mathematical description of growth, denoting the initial cell number and t the time of incubation. Good day, this is Professor Will, CBMD PhD on microbiology, bioinformatics, and computer science. Well, hope you enjoyed listening. Until next time, have a great day.